During the enzyme-catalyzed reaction, the substrate molecules bind to a point on the enzyme known as the activation site. The activation site is specifically shaped so that it can only react with a single substrate. An enzyme-substrate complex is when the substrate binds to the enzyme's activation site. The enzyme will then bind or separate the molecules in the substrate in order to produce certain products. Competitive inhibition is a type of inhibition in which an inhibitor chemical, which is the correct shape, bonds with the activation site of an enzyme and remains there in order to prevent any substrates from binding with the site. Non-competitive inhibition is a type of enzyme inhibition in which a chemical inhibitor bonds to a part on the enzyme which is not the activation site, causing the enzyme to change its configuration, therefore changing the shape of the activation site so that it is no longer receptive to the appropriate substrate. Also demonstrated here is allosteric regulation, in which an inhibitor bonds to a site on the enzyme that is not the activation site, known as the allosteric site. Usually the allosteric regulation opens the activation site that has been closed. This is the inverted process of non-competitive inhibition. Feedback inhibition is a type of enzyme regulation in which a chemical inhibitor will bind to a site on the enzyme that is not the activation site and not change the shape of the enzyme, but block off and restrict access to the activation site, preventing any further reactions. Demonstrated here is denaturing of an enzyme due to temperature. An enzyme can denature due to a number of causes including temperature and pH. Because of how delicate and specific the function of an enzyme is, it can easily be disturbed and denatured if there is a fluctuation in its normal environment.